1 minute. Okay. So, what is chemical bond? The force of attraction that holds the two atoms together. That is called what? Chemical bond. The force of attraction that keeps two or more atoms together in a molecule or compound. That is called what? Chemical bond. So, what is chemical bond? The force of attraction, the force of attraction which holds constituents means atoms, ions, molecule together. Constituents together is called chemical bond. What it is? Chemical bond, right? Now, in it we'll study Lewis symbol. Lewis symbol. Now, what is Lewis symbol? You know, Lewis symbol, basically, it is notation to represent the valence electron in an atom. It is notation to to represent valence electrons, valence electrons in an atom, in an atom. Okay. Now, this notation, they are called Lewis symbol or Lewis notations. Okay. According to Lewis symbol of an element represent the nucleus. Symbol of an element represents hence the nucleus. Now, valence electrons, they are represented by dots or cross around the symbol. Valence electrons are represented are represented by dots or dots or cross around the symbol, around the symbol, around the symbol. For example, atoms like lithium, beryllium, boron, carbon, nitrogen, oxygen, fluorine, neon, right? Now, what is meant by valence electron? Electrons present in the outermost shell, they are called valence electron. Now, valence electrons. Lithium consists one valence electron, beryllium 2, boron 3, carbon 4, nitrogen 5, oxygen 6, fluorine 7, and neon 8. Leuven 8. Okay. So Lewis representation is what? Valence electrons in an atom. Okay. And they're represented in the form of dot. So we'll write Lewis symbol. Lewis symbol. Lithium single dot. Okay. Beryllium 2 dot. Then little bit I draw below beryllium 2 dot. Then boron 3 dots. Then carbon 4 dots. Then nitrogen 5 dots. Then oxygen 6 dots. Then fluorine 7 dots. Then neon 4 pairs like this. So this is what V symbol. So if you see properly, the valency of lithium, beryllium, boron, and carbon are 1, comma 2, comma 3, and 4 respectively. Respectively. On the other hand, valency of nitrogen, comma oxygen, and fluorine are 3, comma 2, and 1 respectively. 1 respectively. Respectively. What is valence electron? Number of electron present in the outermost shell of an atom of an element, that is called valence electron. Number of electrons present in the outermost shell of an atom of an element is called valence electrons. Valence electrons. Now, what is valency? Valence is nothing but combining capacity of an element. Valence is nothing but combining capacity of an element. Valency is nothing but combining capacity combining capacity of an element. For example, nitrogen combined with 3 hydrogen. So valency of nitrogen is 3. But 3 hydrogen combined with 1 nitrogen. So valency of hydrogen it is 1. Similarly, you know, 2 hydrogen combined with 1 oxygen. So valency of hydrogen is 1 and that of oxygen is 2. Similarly, HCl. So valency of hydrogen is 1 That of chlorine is also 1. 1. Okay. So what is valency? Combining capacity of an element. That is called what? Valence. That is called what? Valence. Is there any doubt to anybody regarding these acid symbols?
No, sir.
Okay. Okay. If no doubt, then we'll move to next point. Next point. So next point from this chapter is covalent bonding. Covalent bonding. Now, what is covalent bond? Suppose there are two friends. One is Ram, second one is Sham. If Ram and Sham they went for a party, both they went for a party. Okay. Then if Ram as well as Sham contributes 500 rupees each. How much? 500 each. Maybe it happens Ram will eat more than Sham, or vice versa. That is, Sham will eat more than Ram. Correct? But contribution is what? Equal. Contribution is equal. And such a equal contribution, that is called mutual contribution or equal contribution. Mutual contribution or equal contribution. Now, suppose if you want to prepare HCl, then hydrogen 1 electron combined with chlorine 1 electron. So you'll get HCl molecule. What will get? HCl molecule, right? So the bond which is formed by equal or mutual sharing of electron, that is called covalent bond. The bond which is formed by equal or mutual sharing of electrons is called covalent bond. What it is called? Covalent bond. The bond which is formed by equal or mutual sharing of electron, that is called covalent bond. So bond which is formed by equal or mutual sharing of electron, that is called what? Covalent bond. Formation of HCl, hydrogen contributes 1 electron, chlorine contributes 1 electron, and thus it is equal or mutual sharing of electron. Equal or mutual sharing of electrons, right? Equal or mutual sharing of electrons. So the bond which is formed by equal or mutual sharing of electron, that is called what? Covalent bond, right? Now, atom involved in covalent bond formation contributes equal number of electrons for sharing. The shared electrons become common to both the atoms, that is hydrogen as well as chlorine. If the atom shared one pair of electron, the bond form is called single covalent bond. If if mm, single covalent bond, single covalent bond. What is single covalent bond? When atoms share one pair of electrons, when atom shares one pair of electrons, the bond form is called, the bond form is called single covalent bond, single covalent bond. For example, for example, in a chlorine, one electron combines with one electron from another chlorine and you get a chlorine molecule. That is single bond is formed. The next, a double covalent bond or double bond, double bond when atoms share two pair of electron, when atom share two pair of electrons, two pair of electrons, it is called double bond. For example, oxygen, one oxygen has two electrons, combines with another oxygen having two electrons, we get oxygen, double bond oxygen. That is O2 molecule, right? Then next one is triple bond. When atoms share three lone pair of electrons, when atom share three pairs of electrons, it is called triple bond. What it is called? Triple bond. Okay. For example, in a nitrogen, it has three lone pair of three electrons in the outermost shell. So one nitrogen atom bonded with another nitrogen atom, and it forms a N2 gas. It forms N2 gas. Is there any doubt? The next, covalency. Covalency. What is covalency? Number of electron pair shared by an atom or number of electrons contributed by an atom for the formation of a covalent bond. That is called what? Covalency. Number of electron pairs shared by an atom form a covalent bond. That is called what? Covalency. Number of electrons shared by an atom for formation of covalent bond, for formation of covalent bond is called covalence. What it is called? Covalence. It is called covalency. Is there any doubt? Any doubt? Is there any
Sir,
doubt?
yes, sir, but it's not related to the topic. SPDF is going to be taught or not because it is in one of the question papers.
What is the question, beta?
एक minute. I'll just hold. I'll tell you in 1 minute. हाँ सर, पेपर 2014 क्वेश्चन नंबर 44. Electronic configuration of an element is 1s2, 2s2, 2p6, 3s2 and 3p6. What will be its valency? So this type of question is there. So is SPDF going to come or not?
Read it again.
The electronic configuration of an element
Hmm.
is 1s2, 2s2, 2p6, 3s2, and 3p6. What is its valency?
See,
Three.
what is valency? Electron present in the outermost shell. Now, in this case,
Yes.
third cell is the outermost shell, right?
Yes.
So, how many electrons are present in third cell? Whatever rest to I wrote, that is nothing but electrons. So, third shell,
Eight.
how many
Eight.
electrons?
2 plus
Eight.
6, 8.
Eight, right?
Yeah.
So, valency is equal to number of electrons in
In the,
outermost shell,
yeah.
outermost shell, or valency is equal to 8 minus number of electrons in outermost shell. In this case, till 4, okay, till 4, valence electron is equal to valence. And after 4, 8 minus number of valence electrons. So 8 minus number of valence electron 8. So valency is 0. Is there any doubt? Is there any
Understood,
doubt?
sir. Understood.
Understood? Yes or
Yes.
no?
Yes.
Okay. Okay. So let's come here. Here. Okay. So in this case, I'll make one more column. Valency of lithium is 1, beryllium is 2, boron is 3, carbon is 4. From nitrogen onwards, 8 minus valence is equal to what? 8 minus number of valence electron. So 8 minus 5, क्या आएगा वैलेंसी 3. 8 minus 6, 2. 8 minus 7, 1. 8 minus 8, 0. समझ में आया अभी? Yes or no? Students? Okay, okay. Now, next part is polar covalent bond. Polar covalent bond. Okay, now consider HCl molecule. H combined with Cl forming what? HCl molecule, right? Means the bond which is formed by equal sharing of electron, that is called what? Covalent bond, right? So HCl bond is covalent bond. But after formation of bond, chlorine is more electronegative. Chlorine, it is more electronegative. So it pulls shared pair of electrons of hydrogen towards itself. Shared pair of electrons of hydrogen towards itself. As a result, hydrogen becomes electron deficient, acquires positive charge, and chlorine becomes electron rich, acquires a negative charge, and such a bond which involves charge separation that is said to be polar covalent bond. Right? So, what is polar covalent bond? The bond which is formed by equal sharing of electrons, but after the formation bond, the atom which is more electronegative, it pulls shared pair of electrons of other atom. As a result, positive and negative charges are developed, and the resulting bond is called what? Polar covalent bond. So, the bond which involves separation of charges separation of charges due to higher electronegativity, higher electronegativity of one of the atom, the word atom, is called polar covalent bond. Polar covalent bond. For example, bond in HCl. Similarly, second one is non-polar covalent bond. What is non-polar covalent bond? Let us consider formation of hydrogen molecule from two hydrogen atom. Hydrogen, its atomic number is 1. Its atomic number is 1, right? So, number of electrons are 1. So, hydrogen having 1 electron combines with another hydrogen having another electron and it forms hydrogen molecule. What it forms? H2 molecule, hydrogen molecule, right? In this case, electronegativity of both the atoms, that is, both are hydrogen atom. So it is exactly same. So shared pair of electrons is not pulled by any one of the atom. Okay, and there is no formation of a charges takes place, and such a bond is said to be covalent bond. So the bond which do not involve which do not involve charge separation, charge separation due to similar electronegativity, electronegativity between two joining atoms, between two joining atoms, it's called non-polar covalent bond. Best example, bond between two chlorine atoms, bond between two bromine atoms, bond between two iodine atoms. That is non-polar covalent bond. Is there any doubt?
No, sir.
The next, orbital overlap concept. Orbital overlap concept. Next point is orbital overlap concept. Now, there are 4 different types of orbitals. How many? 4 different types of orbitals. So next part, types of orbitals. So there are 4 different types: s orbital, p orbital, d orbital, and f orbital. Now, s orbital has a maximum capacity to accommodate electron is 2. P orbital it is 6. Then d orbital it is 10. F orbital it is 14. F orbital how much? 14. Right. Now, what are the shapes of orbital? shapes of orbitals. First one is S orbital. S orbital, it is spherical in shape. So this is x-axis. This is y-axis. This is z-axis. Okay. Then second, shapes of p orbital. P orbitals. P orbitals, they are further classified into three types. Px. Px means what? Their lobes, they lie along x-axis. So this is the x-axis, this is y-axis, this is z-axis. So this is px orbital. Then second, py orbital. So in case of py orbital, lobes, they lies along y-axis. And this is what? Z-axis. The next one is shape of pz orbital. So this is x-axis, y-axis. This is z-axis. So lobes, they lie along z-axis. Along z-axis. Is there any doubt? Is there any doubt?
Sir, what is the difference between shells and orbitals?
Uh, Cells, they are denoted by number 1, 2, 3, or letters K, L, M, N. Okay. And orbitals, they are denoted by S, P, D, F. Right. Now, how to define orbit and shell? Orbits, they were also known as shell. Orbit, that is also called as what? Shell. So, orbit is a three-dimensional space around the nucleus where probability of finding of electron is maximum. What is orbit? That is orbit K, L, M, N, orbit or shell. So, orbit is what? Max three-dimensional region where probability of finding an electron is maximum. And what is orbital? Orbital is a space that describe region around an atom where electrons are most likely to be found. Okay. So orbit is three-dimensional space where probability of finding an electron is maximum. But what is orbital? Orbital or subshell? Orbital or subshell is the region around the nucleus of an atom where electrons are present. Where electrons are present. Okay, that I'll write. Orbit or shell. So it is three-dimensional space around the nucleus where probability of finding electron is maximum. Okay. So there are four types of cell: K shell, L shell, M cell, then N cell. So for K shell, N has value 1. N is called actually principal quantum number. L shell, N is equal to 2. M cell, N is equal to 3. and n cell n is equal to 4. Right? The next orbital or subshell, So it is a region around the nucleus, region or space around the nucleus, where electrons are most likely to be present, where electrons are most likely to be present. Is there any doubt?
No, sir.
Okay. So this is what about orbits. Now while writing electronic configuration, hydrogen, atomic number is 1, so electronic configuration 1s1. Electronic configuration. What is electronic configuration? Distribution of electrons amongst various cells and subcells. Shells means 1, 2, 3, 4, etc. Subcells means SPDF, etc. Hydrogen electronic configuration is 1s1. This indicates hydrogen consists of one electron in outermost shell. Then helium 2. So it is 1s2. This indicates there are 2 electrons in outermost shell. Then lithium 3, 1s2, 2s1. So in case of lithium, second shell is there. So 1 electron, second cell is the outermost shell, 1 electron in outermost shell. Then beryllium 4, electronic configuration is 1s2, 2s2. So it consists of 2 electrons in outermost shell. How much electrons? 2 electrons in outermost shell. Then boron 5. So 1s2, 2s2, 2p1. So there are 3 electrons in outermost shell. Then carbon atomic number is 6. Electronic configuration 1s2, 2s2, 2p2. So carbon consists 4 electron in outermost shell. Then nitrogen 7, 1s2, 2s2, 2p3. So 2 plus 2, 4. 4 plus 3, 7. So nitrogen consists 7 electrons in outermost shell. 7 electrons in outermost shell. Then oxygen atomic number is 8. So its electronic configuration is what? 1s2, 2s2, 2p4. So outermost shell is second cell. So it consists of in case of nitrogen, there was a mistake. Actually, there are 5 electrons. By mistake, I wrote 7 electrons. So 5 electrons. In this case, 6 electron in outermost shell. How it is 2 plus 4? 6, 2 plus 3, 5, 2 plus 2, 4 electron, likewise. Then fluorine 9, 1s2, 2s2, 2p5, 2p5. So it consists of 7 electrons in outermost shell. Okay, likewise. Then neon 1s2, 2s2, 2p6. So 6 plus 2, 8. So it consists of 8 electron in outermost shell. So you have to count only electron present in the outermost shell and not innermost shell. Is there any doubt to anybody up till this?
Sir, what is the power?
What
The
is the
S2,
ah,
S2, P4.
1s raised to 2? That power is nothing but number of electrons.
Okay.
Power is nothing but number of electrons. Okay.
Yes,
Student,
sir.
just I'll be there within a minute. Okay. Please wait. Just minute. Okay. Next part. That is orbital overlap concept. Right. We study there are 4 types of orbitals. What are those? S orbital, P orbital, D orbital, and F orbital. Right? Right or not? So they leads to the formation of 2 different types of bond. Okay. So one is called sigma bond and second one is called pi bond. So depending upon the extent and type of overlapping, the covalent bond may be of 2 types. They are sigma bond and pi bond. So next one is orbital overlapping concept. Orbital overlapping concept. Orbital overlapping concept. So depending upon amount of overlapping and type of overlapping, right? Whether the overlapping is maximum or minimum. Maximum overlapping, stronger is the bond. Minimum overlapping, weaker is the bond, right? Type of overlapping means whether the overlapping takes place end to end or axial or lateral. Depending on that, stronger or weaker bond is formed, right? So orbital overlapping concept depending upon the extent and type of overlapping, the covalent bond are of two types. First, sigma bond symbol is like this, and second one is pi bond symbol is like this, right? Now, what is sigma bond? The bond formed by axial overlapping or end-to-end overlapping of half-filled atomic orbital along the internuclear axis. That is called what? Sigma bond. Covalent bond formed by the covalent bond formed by an axial overlapping or end-to-end overlapping of half-filled orbitals along the internuclear axis, along the internuclear axis is known as sigma bond. What it is? Sigma bond. The electron involved in the sigma bond formation are called sigma electron. Electrons involved in sigma bond formation are called sigma electrons. What they are called? Sigma electrons. Single bond present in all the molecule, they are of always of sigma type. Single bond present in all molecules are always sigma type. Sigma bond, it is formed by any one of the following combination, that is SS overlapping, SP overlapping, or PP overlapping. Sigma bond is formed by any one of the following combination What is that? A is SS overlap or overlapping. Second, SP overlapping. And third, PP overlap. Now we'll begin with first SS overlap. First, SS overlapping. Now whenever one s orbital, which is half filled, means contain one electron, overlaps with another one s orbital, what we'll get is SS overlap, nothing but sigma 1. Okay. So this leads to the formation of one sigma bond. Second, sp overlapping. Of course, best example is formation of hydrogen molecule from hydrogen atoms. sp overlapping, formation of HF molecule. When one s half-filled orbital overlaps with one half-filled p orbital, it is called sp overlap. What it is called? sp overlapping. Whenever half-filled one s orbital overlaps with half-filled p orbital, that is called what? sp overlap. What it is? sp overlap. And it takes place end to end. So in sp overlapping, one s orbital of hydrogen, one s orbital of hydrogen, okay, combines with one p orbital of fluorine. This what? This is s orbital of hydrogen combined with p orbital of fluorine. So what do you get? You get one sigma 1, nothing but HF bond. Nothing but HF bond. Then third one is PP overlapping. PP overlapping. Now, whenever one p half-filled orbital of one atom overlaps with one half-filled p orbital of another atom, PP overlapping takes place when one half-filled p orbital of one atom overlaps with half-filled p orbital of another atom, PP overlapping takes place. PP overlapping takes place. For example, formation of F2 molecule. Formation of what? F2 molecule. Formation of F2 molecule. So, one minute. So, this is p orbital, a half-filled p orbital of one atom. This is half-filled p orbital of another atom, right? So, p orbital of fluorine atom, then this is another p orbital of another fluorine atom, and it gives what? It gives a fluorine molecule. This is one sigma bond. One sigma bond. See, in all three cases, SS overlapping, SP overlapping, PP overlapping, we have seen what we see. That is, they overlaps end to end or axially, and thus that leads to formation of a sigma bond. Formation of sigma bond, right? The next pi bond, covalent bond formed by sidewise or lateral overlap of half-filled atomic orbital, that is known as pi bond. Okay, covalent bond formed by the sidewise or lateral overlap of the half-filled atomic orbitals is known as pi bond. Okay. So pi bond is formed by sideways or lateral overlapping. Pi bond is formed by what? Sideways or lateral overlapping. Pi bond is formed by sideways or lateral overlapping. Okay. Electron involved in pi bond formation, they are called pi electrons. Electrons involved in pi bond formation are called pi electrons. What they are called? Pi electrons. Electrons involved in formation of a pi bond, they are called pi electrons. Generally, pi bond is always present in the molecule having multiple bonds, that is double or triple bond. Pi bond is always present in a molecule having multiple bonds that is double or triple bonds. Double or triple bonds. Okay. Then next, strength of sigma and pi bond. Next part is strength of sigma and pi bond. Okay. Generally, sigma bond is stronger than pi bond. Sigma bond is stronger than pi bond. Okay. Because first, sigma bond is formed by axial or end-to-end overlapping. Sigma bond is formed by an axial or end-to-end overlapping. End-to-end overlapping. Okay. And therefore, second reason, overlapping region is large. That is, overlap takes place in a maximum extent. So, maximum overlapping takes place. And therefore, third point, greater is the overlapping, greater is the overlapping, stronger is the bond, stronger is the bond. Okay. On the other hand, pi bond is weak and pi bond is weaker than sigma bond because first, pi bond is formed by lateral or sideways overlap. Pi bond is formed by lateral or sideways overlapping. Pi bond is formed by lateral or sideways. Lateral or sideways overlapping. Pi bond is formed by sideways or lateral overlapping. Second point, therefore, minimum overlapping takes place. Minimum overlapping takes place. Due to minimum overlapping, okay. Third point, smaller is overlapping, weaker is the bond. Smaller is the overlapping, weaker is the bond. Weaker is the bond. Then last point we'll study, that is distinguish between sigma and pi bonds. From this, of course, dipole moment is one more point left, and that will complete. So next one is what? Distinguish between sigma and pi bond. What we have to see? Distinguish between sigma and pi bond. Next topic is distinguish between sigma and pi bond. Sigma and pi bond. Distinguish between sigma and pi bond, right? So we'll write here sigma bond, here pi bond. Okay, now what is distinguish? This is formed by axial or end to end overlapping. This is formed by lateral or sideways overlapping. Lateral or sideways overlapping. Then next, in case of a sigma bond, overlapping is maximum. Overlapping is maximum. Overlapping. It is maximum. In case of pi bond, overlapping is minimum. Minimum. As a result, one minute. Okay. As a result, Sigma bond is strong bond. Then pi bond is weak bond. Pi bond is weak bond. Is there any doubt to anybody? So this is what I.
Sir, how does a pi bond looks like?
What? How does pi bond?
Looks like.
Okay. Pi bond formation we started, but I didn't show you. Okay, I'll show you. Right. For example, one minute, I'll show you. So what a p orbital of one atom that is having dumbbell shape. This what p orbital of another atom. Right. They overlaps sideways. And they forms a pi bond. So this is what the diagram shown. Okay, there is a formation of a pi bond. Is there any doubt?
So there is only one overlapping.
Yeah, two overlap. This is also overlap, no?
No,
One
like this
overlap.
is overlapping with.
Correct. One overlap, half overlap of one atom, another half overlap of another atom. Okay,
Okay, so in pi bond, only PP overlapping is there.
right. And sigma bond, it has independent existence. Independent existence. However, pi bond is always present with sigma bond. Pi bond is always present with sigma bond. That is, it do not exist independently. For example, carbon single bond, right? You get one sigma bond. But carbon double bond will get one sigma and one pi bond. So this is sigma and this is pi. Then in carbon triple bond, you'll get one sigma and two pi, right? So you can say this topmost is sigma, west way. Topmost will be considered as pi bond, downmost pi bond, and this middle one, nothing but sigma bond. So pi bond is present only along with sigma bond. You do not have separate existence. You do not have separate existence. कोई doubt है?
No, sir.
Then last part from the chapter. Dipole moment. What is that? Dipole moment. Dipole moment. What is dipole moment? Dipole moment is a product of charge and distance between the center of cation and center of anion. Dipole moment is the product of charge and distance between center of positive and negative charge. Dipole moment. First, it is denoted by letter mu. 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 Okay, mu. Then it is a product of, it is product of magnitude of charge, magnitude of charge, and the distance between and the distance between the centers of positive and negative charges. It is a product of magnitude of charge and the distance between the centers of positive and negative charges. Okay. So mathematically, it is expressed as, it is expressed as dipole moment mu is equal to charge q into distance of separation, that is r, and therefore mu is equal to q into r. Mu is equal to what? Q into r. Dipole moment is usually expressed in d by unit. It is expressed in moment is expressed in d by unit right? Then 1 d by, that is equal to 3.33564 into 10 raised to minus 13 centimeter. That is cm, not centimeter exactly, coulometer. Okay. Now, for remembrance purpose, you can, but C stands for what? Where C, coulomb, you can say quantity of charge. And m, meter. M is what? Meter. Okay. Now, dipole moment is a vector quantity and it is expressed by small arrow with the tail pointing towards positive center and head pointing towards negative center. So, dipole moment. Have you studied scalar quantity and vector quantity?
Yes, sir.
So, what is scalar quantity and what is vector quantity? Yes. What is scalar quantity and what is vector quantity? Tell me. Vector quantity has both magnitude as well as direction. However, scalar quantity, it has only magnitude. It do not have direction. It do not have direction. It do not have direction. Okay. So dipole moment is a vector quantity. What is vector quantity? Quantity which have both magnitude as well as direction. That is called what? Vector quantity. Okay. Is there any doubt? Yes or no? Students? Are you understood vector quantity or not?
Yes, sir.
So, dipole moment is a vector quantity. Vector quantity since it has both magnitude and direction. Okay. Dipole moment is represented by arrow is indicated by arrow with with tell towards positive center positive center and head pointing towards negative center. Head pointing towards negative center. Best example is HF. Now, fluorine is more electronegative than hydrogen, and therefore, tail is pointed towards more electropositive atom, and head is pointing towards more electronegative atom. More electronegative atom. So, HF, hydrofluoric acid, whenever there is electronegativity difference, then dipole moment exists. When the two joining atoms they are having same electronegativity, dipole moment does not exist. Okay, then linear molecule like beryllium fluoride or CO2 have zero dipole moment. Okay, so beryllium is at center, fluorine. Now, fluorine is more electronegative. So, vector is pointed towards fluorine. As a result, whatever dipole moment created in one beryllium fluoride bond, it is cancelled by another. And that's why net dipole moment, nothing but mu, that is zero. Similarly, carbon dioxide. Oxygen is more electronegative than carbon. So their electronegativity becomes zero. Okay. Is there any doubt? Is there any
No,
doubt?
sir.
Follow to all students. Array, you are answering. Okay. Then, a last topic from biology. What is that? Biotechnology. Biotechnology. Do you know anything about biotechnology?
Sir, there are small implants.
Small? Correct. Whatever you are saying is correct. Small?
So there are small implants. I was muted, sorry. There are small implants that are implanted mainly in humans and sometimes animals, and basically in humans and animals to manipulate the neurological activity. It's based on biology and technology, so it's biotech. So I thought.
Okay, correct. ठीक है. Now, what is biotechnology? Biotechnology is a combination of two words. Biotechnology. It is a combination of two words. What is that? Bio deals with biology, and technology means technology. Technology. So these two words, they are used to solve problems, improve lives, and protect the planet. So biotechnology is a branch of biology that deals with use of technology to living organism to solve the problems, improve lives, and protect the planet. Actually, I wrote in wrong way. Planet means entire planet. I wrote by mistake plant. So, what is biotechnology? It is a branch of biology that involve application of technology in utilizing the living organism or their parts to produce products useful for humans. So biotechnology or biotechnology is the branch of biology, is the branch of biology that involves application of applications of technology in utilizing the living organism or their parts to produce products useful for products useful for human. Now, What are the application of biotechnology? Applications. First, it is used in agriculture. Agriculture to produce improved varieties of plants and animal breeds. To produce improved variety of plants and animal breed, which is disease resistant, which is disease resistant. Okay. Second, food processing. Food processing. Biotechnology, it is used in food processing industry to produce additives. Produce additives. The next, biotechnology, it is applied to produce biocatalyst. Biocatalyst. Similarly, to produce biopolymers like polylactic acid. Then biotechnology, it is used to control pollution. Pollution. Then it is used to treat wastewater. Treat wastewater. Then it is applied in the field of healthcare to produce several vaccines, drugs, we can say pharmaceuticals. Pharmaceuticals. It is used to produce vaccines, drugs, and hormones like insulin, etc. etc. So, what are the applications of biotechnology? Biotechnology is used in agriculture. Where? Agriculture. To produce improved variety of plants and animal breed, which is disease resistant, which produces more fruits, more yields, etc. etc. Okay. Then second, in food processing industry, to produce additives, then to produce biocatalyst, then to produce biopolymer, to control pollution, to treat wastewater, then it is using pharmaceutical, that is vaccine, drugs, hormones like insulin, etc. Okay, so biotechnology in agriculture and health we finished. Then methods used in biotechnology will study. What we'll study? Methods used in biotechnology. Next part is methods used in biotechnology. Methods used in biotechnology. Methods This is small topic methods used in biotechnology. Okay. So there are different methods they were used in biotechnology, genetic engineering, tissue culture, then bioinformatics. Okay. Genetic engineering. Next, tissue culture. And next, bioinformatics. Bioinformatics. Now, genetic engineering means what? Like recombinant DNA technology and gene editing. Genetic engineering involves genetic engineering involves gene editing, gene editing and recombinant DNA technology. Recombinant DNA technology. Recombinant DNA technology. Okay, now what is recombinant DNA technology? It involves using enzyme like restriction enzyme, DNA ligase to cut, join, insert gene from one organism into another, creating new genetic combination. Now, gene editing. Gene editing involves a CRISPR-Cas technique for precise modification of an organism's DNA. Then, what is tissue culture? Tissue culture. The process of growing cells, tissues, or organs in a controlled laboratory environment on a nutrient medium. The process of growing cells, comma, tissues, or organs in a controlled laboratory environment meant on a nutrient medium, on a nutrient medium. Okay. Now, cell culture techniques similar to tissue culture, but focus is specifically on growing cells. Then, what is plant biotechnology? Plant biotechnology includes methods for improving crop varieties like tissue culture, genetic modification for traits, etc. etc. The next, bioinformatics. Bioinformatics. The use of computational tools to analyze and manage large biological data sets such as DNA and protein sequences. That is called what? Bioinformatics. use of computational tools to analyze and manage large biological data sets such as DNA and protein sequences, such as DNA and protein sequences. That is called what? Bioinformatics. Now, bioinformatics that is also based on protein engineering, that is modifying the properties of protein to create a new or improved functions. New or improved functions, right? New or improved functions. So, is there any doubt about this?
No, sir.
So, what are the method of biotechnology? First one is genetic engineering. Second one is tissue culture. Third one is bioinformatics. Okay, there are several methods, but only these three, they are included in your syllabus. Okay. Generally, genetic engineering and tissue culture technique, they are used in biotechnology. Genetic engineering allows for manipulation of DNA to introduce new traits. Traits means characters, while tissue culture involves growing cells or tissues in controlled environment. Growing cells or tissues in controlled environment. Okay. कोई doubt है? So, what are three important techniques in biotechnology? First, genetic engineering, recombinant DNA and gene editing. Then second, plant tissue culture. And third, bioinformatics, nothing but transgenic, genetically modified organism. Genetically modified organism, right? Now, genetic modification is a method in which biotechnology is specifically designed to alter existing strands of a DNA within the living organism, and this process involves techniques that allows for the addition, removal, alteration. Alteration means changes of genetic material, thereby enabling the manipulation of an organism trait. Right. Basically, there are different tools used in biotechnology, like transfection of electro proportion, then chemical mediated transfer, then micro injections, gene gun. So these are the various techniques. And of course, there are around 10 branches of biotechnology. Whenever biotechnology is used in medicine, it is called medical biotechnology. Whenever biotechnology is used in agriculture biotechnology. Whenever biotechnology it is used in industry, it is industrial biotechnology. Whenever biotechnology is used in environment, it is called environmental biotechnology. Biotechnology, whenever used in forensic, it is called forensic biotechnology. Then biotechnology used in genes, that is called genetic engineering. Then bioprocessing is there. bioinformatics is there. Bioinformatics is there, right? Is there any doubt? Is there any doubt? Students, are you listening my voice or not?
No, sir.
No, sir.
Is there any doubt?
No, sir. No, sir.
Okay, okay. Okay, now, what is basic principle of biology? Sorry, biotechnology. Biotechnology, they are based on genetic engineering and bioprocess engineering, right? So principle of biotechnology, principle of biotechnology, single L, are what? Genetic engineering, and bioprocess engineering. Okay. Genetic engineering involves manipulation of an organism's DNA to alter its trait. Genetic engineering involves manipulation of an organism, manipulation of an organism DNA to alter its trait, to alter its trait means characteristics. Okay. Then bioprocess engineering is used on a large scale cultures of microorganisms to manufacture specific biotechnological product. Okay, bioprocess engineering involves specific biotechnological product, manufacture of specific biotechnological product, biotechnological product. Point out.
No, sir.
Okay, there are four main areas of biotechnology, that is medical, agriculture, marine, okay, industrial. So areas of biotechnology. First, medical. In medical field, you can say. Second, industrial. Then third, agriculture. And last one is marine. And fourth one is marine. Okay, these are the four areas. Okay, and this is what we studied biotechnology. Right. Now, is there any doubt to anybody?
No, sir. No doubt.
Everything is clear?
Yes, sir.
Then, your test, they are going to be start from Friday onwards, right or not? 1 day gap is providing so that you'll study. Understood? And whatever doubts are there, you may feel freely and ask it, and please study more. I want at least 20 students should qualify first level. How many students? Minimum 20. Understood or not? Are you listening my voice? Yes or no?
Yes, sir.
Okay, and the student will get 100 out of 100 marks. Okay, I'll give a cash price of 2000 rupees, whatever may be the number. Understood? Understood or not?
Yes, sir.
Okay, take challenge and go ahead. Wish you all the best. We'll stop today.
Bye, sir. Thank you, sir.
Okay, bye. See you. Yes.